Hello class and welcome back. So today we get to do osmosis diffusion, transportation, and a little bit more about cell biology. So in today's PowerPoint, I'm going to go over the movement of materials, and then I will talk a little bit about the different activities that you have and practical. So the key points for today: how do cells regulate their environment? How do our bodies use fluid gradients to maintain homeostasis? Basic structure of a cell. So knowing that there's the plasma membrane and that cytoplasm is containing organelles, the nucleus is the main powerhouse, and that's where everything, the brain of it. So the function of cells, metabolic and energy use, synthesis of molecules, communication, reproduction, and inheritance, transportation. So how does that all get there? I went through that previous stuff really quickly because, well, it's something we talked about last week. So there's a fluid mosaic model. The proteins like icebergs floating in the ocean. So separation of intracellular and extracellular materials. This is done by the plasma membrane itself. It allows for things to go in and out of it at specific times. The membrane has some potential. Ions concentrate on either side. The hope is always to make it so that there's a homeostasis is going on. So if one side is positive, the other one should be negative. This is important for transportation. So you have the phospholipids, and here's the bilayer sheets of those. Cholesterol, proteins, carbohydrates, fluid mosaic model. Okay, so you have the glycosyl, which is a combination of carbohydrates and lipids. You have the proteins and the outer surface, membrane lipids. Okay, scroll through that, sorry. Okay, so you have a bilayer of the phospholipids, which, let's see, right here and right here. And you can see that there are polar heads, both the interior and the exterior of it. Nonpolar tails facing each other on the interior of the membrane, which are these right here. Hopefully you can see that marking. And there are cholesterol little bundles kind of situated all throughout, just kind of interdispersed. The amount of these is determined by the fluid nature of the actual membrane itself. The fluid nature provides, and it also allows for the distribution of molecules within the membrane itself. So that would be through there. The phospholipids automatically reassemble if a membrane is damaged. The membrane can fuse with each other. So the membranes themselves can kind of repair themselves just a little bit. Kind of cool. Let's erase all this. Sorry. membrane proteins. Integral or intrinsic. They both extend deeply into the membrane and often exist from one surface to the other. There are some crazy little channels that you'll be able to kind of see happening. The peripheral and extrinsic attached to the integral protein at either of the inner or outer surfaces of the lipid bilayer. Okay, so movement through the plasma membrane. The diffusion. This is when the smaller polar or nonpolar molecules such as O2 and CO2 have a constant gradient. So diffusion in real life is kind of like a coffee. You put coffee in water and it diffuses throughout it, or tea, or an air freshener, that kind of thing. So osmosis is diffusion of water molecules. So this kind of shows you the difference in the type of diffusion. You have a simple diffusion, then facilitated diffusion, which is through a channel, and the facilitated diffusion carrier. Mediated transportation, this is a facilitated diffusion, active transport, and secondary active transport. Okay, this table right here will be your best friend. You will see it again, I promise, on your practical, on your quiz. Very good possibility. So normal diffusion is passive. It goes from high to low gradients. And so no membrane is needed or semi-permeable membrane. It does not require energy. Examples of this is water, gases, and steroid hormones. Facilitated diffusion is passive. Osmosis is also passive. The only one that's actually active, and you can see that the direction of movement in the active is different. It goes from low to high instead of high to low. These all need a way through which the molecules can allow themselves to just kind of permeate through. However, with active transport, you need a protein pump and ATP. So energy is used. Remember, osmosis is only water. Okay. You're doing kind of actual lab stuff this week. Kind of not. I'm just going to skip through it because it's not quite the same. So, hypotonic, water always moves from where it is most concentrated or has less solution to where it is less concentrated. Hypotonic solutions have a lower concentration of solutions compared to other solutions. More solutes within the cell. The cell inflates or bursts because of the water in the high concentration outside the cell. Hypertonic, on the other hand, have a higher concentration of solutes, less solutes within the cell, so this cell starts to shrink or cryonate. And because a higher concentration of water inside the cell, isotonic is the balance or homeostasis. So a 5% glucose solution and a 0.9% normal saline solution serve as an isotonic solution to the human cells. Try to figure out what exactly is happening. So you have, these are regular blood cells. Isotonic, hypertonic, and hypotonic. Okay, so this week you have 4 lab activities that you need to complete. Those are found on McGraw-Hill Connect through my lab section. So they should be kind of interesting and fun, hopefully. McGraw-Hill Connect is kind of touchy, so be prepared to spend a decent amount of time on them. I will be putting up a study guide and or study Kahoot for your practical, which will be next week. It will be 1 hour. There are 50 questions/stations with 2 parts each. So 100 questions, 100 points. Okay, I hope you guys have a great week. If you have any questions, please let me know. As far as grading is concerned, please wait until Tuesday after I've graded to send me emails about not getting something right. I go over everything on Tuesdays when I'm grading. Okay? Talk to y'all later. Cheers.